have you ever stopped and wondered, why you look the way you do? Like why you have your mom's eyes, maybe your dad's nose, or why certain health things seem to pop up in your family. It all comes down to this amazing, really intricate story. The story of genetics. Though sometimes you do wonder why that family trait decided to stick around, right? Welcome to the deep dive. We take source material, distill it down, basically give you the essential insights so you can be genuinely well-informed fast. Today, we're diving deep into foundations of molecular biology and genetics. We're pulling insights from an introductory lecture series by Dr. Amer El-Zawili. Our mission is to give you a solid, hopefully engaging grasp of how life's blueprint works and really why it matters to you. Now, you might hear genetics and, yeah, maybe think textbook, dry stuff.
Yeah.
Here's where it gets genuinely interesting. We're talking about the actual code that makes you, well, you. And understanding this, it's not just about curiosity. It's like unlocking the fundamental operating system of life itself. It gives us this incredible power to understand disease, engineer solutions, maybe even shape the future.
That's exactly right. We'll be looking at everything from how specific traits get passed down through generations to the really intricate dance cells do when they divide. Perfectly choreographed, really. The goal is to give you those aha moments, where the dots connect and you see the logic underneath all the biology.
Okay, let's unpack that. Our source material, it asks this really fundamental question right at the start. Why bother studying genetics? And the answers, when you think about it, are incredibly broad. They touch almost every part of modern life. It's quite impactful.
Yeah, what's fascinating is how quickly you see the connection to real-world applications. I mean, across so many critical areas. Think about understanding disease. That's how we trace how conditions are passed down through families. That's huge. But it also massively improves agriculture, helping develop better crops, more resilient ones. And, well, it's a cornerstone of the whole biotech industry. It directly influences how we design new drugs, develop them, even produce them on a large scale. Which raises a pretty important question, doesn't it? How can something so fundamental impact so many different fields?
That is a huge question. It really makes you think beyond just the how something works. I mean, we're talking about the fabric of life, right?
Yep.
So to start putting those impacts together, what are the absolute basic concepts we need to get our heads around first?
Right. Our source defines some key terms. These are really the bedrock of genetics. Let's start with the most basic unit, the gene. Put simply, it's a specific stretch of DNA, and it helps determine a particular characteristic.
Okay, a gene. And these genes, they're not always identical, are they? They come in different versions called alleles, like eye color, right? Different alleles lead to blue eyes or brown eyes, different versions of that eye color gene.
Exactly. And the locus, that's just the specific physical spot on a chromosome where you'd find a particular allele. Think of it like its address.
Got it. Locus is the address. Then there's your genotype. That's what the complete set of alleles you have, your personal genetic blueprint.
Precisely. Your genotype is the blueprint. And that blueprint then determines what you actually see, which is the phenotype or the trait. So phenotype is the observable appearance, what makes you look the way you do or function the way you do.
Okay. Genotype is the plan. Phenotype is the result.
And we can categorize individuals based on those alleles. A heterozygote, that's someone with two different alleles at a specific locus. So maybe one for blue eyes, one for brown.
And the opposite.
That's a homozygote.
Yeah.
Someone with two of the same alleles at that locus. So two alleles for blue eyes, for instance.
Okay, so putting these pieces together, what's the big picture here? It's basically that your genotype, your genetic makeup, dictates your phenotype, your traits. But it's not just a static plan, is it? It's like a dynamic set of instructions. How does that blueprint, the DNA, actually become, well, the proteins and structures?
That's where the central dogma comes in. The information flows. It goes from DNA, gets transcribed into RNA, and then translated into protein. It's this crucial hierarchy, making sure everything gets built correctly.
Right. DNA to RNA to protein, that flow is key. Now, when we talk heredity, passing things down, there's one name that always comes up, Gregor Johann Mendel. His work, often with pea plants of all things, really laid the foundation for everything we just discussed. It's amazing, really, considering he had no idea about DNA.
He was absolutely ahead of his time. Just with careful observation of pea plants, he basically figured out the rules of inheritance long before we knew what was being inherited. Our sources really stress the need to not just understand, but to actually apply Mendel's first principle. That means, solving genetic problems.
Yeah.
Things like monohybrid crosses, dihybrid crosses, and being able to tell the difference between genotype and phenotype in practice. That's where the real understanding clicks in.
I like that, moving from theory to actually doing it. So for those of us picturing pea plants now, what's the core idea behind Mendel's first principle? Segregation.
Okay, the core idea is pretty straightforward. Genes come in pairs. We get one copy from each parent. And when an organism makes its reproductive cells, sperm or eggs, the gametes, the two alleles for each trait, separate or segregate. So each gamete ends up carrying only one of the two alleles.
Okay. They split up. Can you walk us through an example? How does this work in a problem?
Sure. Our source uses a classic example. Imagine you have an F1 plant that's the first generation offspring from crossing two purebred parents. Let's say this F1 plant is heterozygous for seed shape. It has one allele for round seeds, let's call it R, which is dominant, and one for wrinkled seeds, R, recessive. So its genotype is RR. Now, if you backcross this R plant to its purebred parent that only has round seeds, so its genotype is RR, what proportion of their offspring will have wrinkled seeds?
Okay. So R crossed with RR.
Right. If you draw out a Punnett square, you'll see the possible offspring genotypes are RR and RR. Since R round is dominant, both RR and RR plants will have round seeds.
Ah, so none of them will have wrinkled seeds. The
No.
proportion is 0.
Exactly. 0. It shows how understanding segregation helps predict these outcomes and how dominance works.
That makes it really clear. Dominant traits can totally hide recessive ones. Okay, then there's Mendel's second principle, independent assortment. What's the gist of that one?
This one says that alleles for different genes like, say, seed color versus seed shape segregate independently of each other when gametes are formed. Basically, inheriting a specific allele for seed color doesn't affect which allele you inherit for seed shape. If those genes are on different chromosomes or very far apart on the same one, they sort themselves out independently.
Okay, so one trait doesn't drag another along with it, usually.
Pretty much.
And this independent assortment, that lets us predict how multiple traits are inherited together, right? Which gets important for more complex situations.
Absolutely. It scales up our predictive ability.
And all these predictions, they rely heavily on probability, don't they? There are those two basic rules in genetics. If you need the chance of two independent things happening together, you use AD, which means you multiply their probabilities. But if you want the chance of either one or another mutually exclusive thing happening, you use OR and you add the probabilities.
Exactly. And this is crucial for figuring out real-world outcomes. Our source gives a good example with blood types. It asks, if the probability of being blood type A is 18 and the probability of blood type O is 12, what's the probability of being either blood type A or O?
Okay, either means add, so 18 plus 12.
Right. You need a common denominator, so 12 is 48. Then 18 plus 48 equals
58.
58. Simple addition, but it shows how these probability rules give us a quantitative way to predict genetic outcomes. It's not just guesswork.
Okay, so that covers how traits get passed down. But what about just basic growth? How do our cells multiply to build us up, repair damage? That brings us to mitosis. Our sources describe it as this highly controlled process in our regular body cells, somatic cells in eukaryotes, organisms with complex cells.
Mitosis is all about accurate division of the nucleus. The key outcome is producing two daughter cells that are genetically identical to the parent cell, identical copies. It's the fundamental process for growth, development from an embryo, and repairing tissues throughout life. It's happening in you constantly.
And this happens as part of the larger eukaryotic cell cycle, which is divided into interphase and M phase, right? M phase includes mitosis itself and then the cell actually splitting, cytokinesis.
Correct. And interphase is the preparation stage. It has subphases, G1, S, and G2. The crucial thing happens in S phase. That's S for synthesis. The DNA gets replicated. So a cell that's finished S phase and is in G2 actually has twice the amount of DNA as it did back in G1.
Twice the DNA, ready to divide. And what about cells that aren't dividing?
They often just hang out in a resting state called G0.
And the DNA itself, it changes its look, doesn't it? In interphase, it's like long threads, chromatin. But after S phase, in G2, each chromosome isn't just one thread anymore. It's made of two identical sister chromatids joined at the hip, basically, a centromere. And these become visible as those dense rod-like things we picture as chromosomes during M phase.
Exactly. Ready for the division dance. Now, the phases of mitosis itself. It's a very precise sequence. First, prophase and prometaphase. The DNA condenses tightly into those visible chromosomes. The nuclear envelope, the membrane around the nucleus, breaks down.
Then metaphase. Meta for middle.
You got it. The duplicated chromosomes line up neatly right in the middle of the cell. It forms what's called a metaphase plate. Very orderly.
Okay, lined up. Then what?
Anaphase. Ana sounds like a part. The sister chromatids finally separate. They're pulled to opposite ends, opposite poles of the cell. Once they separate, Each one is considered a full-fledged daughter chromosome.
Separated.
And finally,
Telophase. The chromosomes reach the poles, they start to decondense, relax back into chromatin, and new nuclear envelopes form around the two separated sets of chromosomes.
so you have two distinct nuclei forming.
Right, and the very last step is cytokinesis. Cyto for cell, kinesis for movement. The cytoplasm itself divides, usually pinching the cell in two. Result, two separate, identical daughter cells. Wow. It really is like an incredibly choreographed dance, isn't it? The precision needed to make sure each new cell gets a perfect, complete, identical set of instructions every single time. It's kind of mind-boggling.
It really highlights how robust our cellular machinery is.
Okay, so if mitosis is about making identical copies for growth and repair, what about making the next generation? What about reproduction? Our sources point to meiosis for that, a different kind of cell division.
Exactly. Meiosis is specialized. It only happens in the cells that will become reproductive cells, sperm, and eggs. And here's the crucial difference from mitosis. Meiosis produces 4 daughter cells, and they are genetically unique, not identical. Plus, each daughter cell has half the number of chromosomes as the original parent cell. This reduction is absolutely essential.
Half the number. Why is that so important?
Think about fertilization. If sperm and egg each carried a full set of chromosomes, the resulting offspring would have double the correct number. Meiosis halves the chromosome number, reducing it from diploid, 2 sets, to haploid, 1 set, so that when sperm and egg fuse, the normal diploid number is restored.
Ah, okay. So it keeps the species chromosome count stable generation after generation. Makes sense.
So its key functions are 1. Reduce chromosome number from diploid to haploid. 2. Make sure each haploid cell still gets a complete set of chromosomes. And 3, crucially, generate genetic diversity among the gametes.
Generate diversity? How does it do that? Mitosis was all about identical copies.
The unique events happen mostly in meiosis first. It's quite different from mitosis. First, in prophase I, something called synapsis occurs. The homologous chromosomes, the pair consisting of one chromosome from your mom and one from your dad, for each type, find each other and pair up very closely along their entire length. This paired structure, with 4 chromatids in total, 2 sister chromatids per homologous chromosome, is called a tetrad, or bivalent.
Okay, they pair up intimately.
And while they're paired up like this, crossing over happens. This is a physical exchange of genetic material between non-sister chromatids. So, between a chromatid from the mom-derived chromosome and one from the dad-derived chromosome.
Swapping pieces.
Exactly. Little segments of DNA get swapped at points called chiasmata. This shuffles the alleles between the homologous chromosomes, creating new combinations on single chromatids. It's a major source of genetic variation.
Wow. Okay, so that mixing happens right at the start. What then?
After lining up in the middle during metaphase I, but as pairs, not single chromosomes like in mitosis, the homologous pairs separate during anaphase first. One entire duplicated chromosome, still with its 2 sister chromatids, goes to one pole, and its homologous partner goes to the other pole. So the cells at the end of meiosis I are now haploid in terms of chromosome number, but each chromosome still consists of 2 chromatids.
Okay, so meiosis I separates the pairs. Then what's meiosis the second of 4?
Meiosis the second is actually much more like mitosis. The chromosomes line up individually in the middle, and then, in anaphase the second, the sister chromatids finally separate. This results in 4 haploid cells, each with only 1 chromatid per chromosome type. And importantly, thanks to crossing over and the random way pairs separated in meiosis the first, these 4 cells are genetically distinct from each other and from the parent cell.
So putting it all together for you listening, what does this mean personally? It means every time sperm or eggs are made, your genetic deck gets shuffled in a unique way. You get a novel mix of the alleles you inherited from your parents thanks to segregation, independent assortment, and that crucial crossing over step.
Absolutely. And connecting this back, the source material explicitly asks about the difference between meiosis I and mitosis. It's a critical distinction. The key is, in meiosis the the homologous pairs are randomly divided. One whole chromosome from each pair goes to a daughter cell. In mitosis, the sister chromatids separate, so both copies of each chromosome get passed on. That random division of pairs in meiosis the is fundamental to generating genetic diversity. It's the engine of variation, really.
Okay, we've covered how traits are passed down, how cells divide, but how do scientists, like genetic counselors, actually track traits, especially genetic diseases, through real families? Our sources introduce pedigree analysis, which sounds a bit like genetic detective work.
It really is. Pedigrees are basically family trees, but drawn using standardized symbols to map out the inheritance of a specific trait over generations. We analyze the patterns in the chart to figure out the mode of inheritance. Is it dominant, recessive, etc., and try to assign genotypes to individuals.
Mm.
But this raises an important point. What assumptions do we make when we read these charts, especially for conditions that aren't very common?
Right, assumptions. Our source mentions one for traits that are rare and autosomal recessive. Autosomal meaning the gene is on a regular chromosome, not a sex chromosome. The assumption is if someone marries into the family shown in the pedigree, we generally assume they are not a carrier of that rare recessive allele. So we'd assume they are homozygous dominant, AA, unless there's strong evidence otherwise, like if they have an affected child. It just simplifies things because the chance of two unrelated people both carrying the same rare allele is low.
That's a key practical assumption.
Yeah.
And understanding the type of mutation helps interpret pedigrees too. Recessive mutations, often they involve a loss of gene function. These can be null or amorphic alleles, meaning they produce a protein that doesn't work at all, or maybe no protein is made. Or they might be hypomorphic alleles, hypo meaning less, so they produce a protein that works poorly, or just reduced amounts of the normal protein. The effect is often only seen if you have two copies of this faulty allele.
Okay, so recessive is often about losing function. What about dominant mutations?
Dominant mutations frequently involve a gain or change of gene function. Our source mentions hypermorphic alleles, hyper for more, which might cause problems because too much normal protein is made, or the protein is overly active. And then there are neomorphic alleles, neo for new. These produce a protein with a completely new function, or sometimes a protein that actually interferes with the normal wild-type protein's function. That last one is sometimes called a dominant negative effect.
So dominant effects can be quite varied. How does this play out in pedigrees? Let's take autosomal recessive ones first. The key pattern you said is that affected individuals can show up even if their parents are unaffected, because both parents could just be carriers, heterozygotes.
Exactly. And the source gives a strategy for assigning genotypes.
Ah.
1. Label all affected individuals A, homozygous recessive.
Hmm.
2. If unaffected parents have an affected child, those parents must be carriers, so label them A. 3. Unaffected children who have an affected parent must also be carriers because they got a recessive allele from that parent. 4. People marrying in who have unaffected children with a known carrier are usually assumed AA based on that rarity assumption. 5. For individuals where you can't be certain, you sometimes have to calculate probabilities.
Okay, a logical step-by-step process. Now, for autosomal dominant pedigrees, what's the main pattern there?
Usually affected individuals have at least one affected parent. The trait doesn't typically skip generations like recessive traits can. Makes sense, right? If you have the dominant allele, you show the trait. And again, for traits that are rare and dominant, we make another useful assumption. Affected individuals are most likely heterozygous. B using B for the dominant allele now. Why? Because if the dominant allele B is rare in the population, it's unlikely that two affected people would happen to meet and have children. It's much more common to see crosses between an affected person and an unaffected person.
So analyzing these family charts, these pedigrees, along with understanding mutation types, that's how we trace these invisible threads of heredity. It really is like solving a biological mystery.
It is, yeah.
So let's recap. Today, we've journeyed through the core ideas of genetics, from the tiny genes and alleles determining traits, through those incredibly precise cell dances, mitosis for growth, meiosis for reproduction and diversity, all the way to the detective work of pedigree analysis, tracking inheritance patterns. We've seen why genetics is just so vital for understanding disease, boosting agriculture, and really driving the whole field of biotechnology.
And remember, knowledge is most powerful when you understand it and can apply it. These aren't just abstract concepts in a textbook. They are, quite literally, the fundamental language of life itself. Understanding them gives you this powerful lens for viewing the biological world, appreciating its complexity, its elegance, its order.
Yeah, this deep dive really shows how something as incredibly complex as life itself can actually be broken down into rules that are surprisingly understandable. Elegant, even. It really brings out the joy of discovery, I think. So as we wrap up, what stands out most to you? Thinking about the power of genetics, how it lets us predict things, explain things, what kind of future possibilities or maybe even ethical questions come to mind when we get better at identifying specific genetic factors for complex traits or diseases. Maybe think about how understanding these basics could reshape fields we haven't even connected to genetics yet. Just something to ponder as you keep connecting these dots in your own world.